Biology is the oldest technology on Earth. It is a system of complex molecular machinery that has been operating for billions of years. These massive redwood trunks are the result of biological architecture, living systems that autonomously build physical structures on a skyscraper scale, using only sunlight, water, and air. For most of our history, we could only observe these processes or slowly guide them through selective breeding. We were limited by what nature provided. Synthetic biology applies engineering principles to these biological systems, treating the cell as a programmable workspace where we can design specific biotechnologies. After decades spent learning how to sequence or read genetic code, we are beginning to actively reprogram it. While computers run on a binary code of ones and zeros, living systems run on a genetic code of 4 chemical letters, A, T, C, and G. Synthetic biologists use biobricks, standardized DNA sequences that can be reliably snapped together like electronic components on a circuit board. This registry includes instructions for a cell, promoters, ribosome binding sites, coding sequences, and terminators. Connecting these creates genetic circuits that execute logic commands like and, or, and not, identical to logic gates in a computer. Living cells can now process chemical inputs and deliver programmed biological outputs with mathematical precision. At an industrial scale, these biological programs can transform ordinary yeast or bacteria into microscopic manufacturing factories. This is the Artemisia plant. It is the natural source of artemisinin, a critical anti-malarial drug. Previously, the global supply of this medicine was entirely dependent on the yields of this single crop. Microbes can now be programmed to brew the drug's precursors in fermentation vats, providing a reliable alternative to traditional chemical synthesis. This same manufacturing approach produces everything from biodegradable plastics to high-performance industrial materials. Standardization allows us to manufacture complex molecules at an industrial scale with the predictability of a production line. Genetic logic gates enable the creation of interactive, autonomous biological machines that operate directly inside the human body. These living therapeutics are engineered cells programmed to navigate the bloodstream and identify specific pathological cues. These cells can analyze the complex surface of a target, identify specific cancer markers, and release a payload of medicine exactly where it is needed. This logic also applies to the environment. We can deploy bacteria programmed to seek out and consume toxic pollutants, breaking them down into harmless components. We are moving from using blunt chemical instruments to deploying precision, self-guided biological tools. As we learn to design bespoke organisms and wire living circuits from scratch, we have to ask ourselves, what does it actually mean to design life?